So in the vitamins, we have 4 vitamins that are key relevance. That's vitamin A and D, who are both fat-soluble vitamins, and folic acid and B12, who are both water-soluble vitamins. Each of these 4 vitamin groups has its own target group, and in all the 4 cases, we have specific problems with the vitamin. So mostly the problems are undernutrition, and in most cases, we try to solve this by the use of supplements. So I'm going to try to give you an idea what is the basic mechanism for these vitamins, what can we do about it, and how do we see the evolution for the next future. So we start with vitamin A, and vitamin A is already known for many, many years. We know quite well the basic mechanisms on how it actually works and how it can induce specific deficiencies. You can find vitamin A in animal-based foods as well as plant-based foods. The animal-based foods are the best bioavailable, and it's quite logical because you can absorb it quite well, either from liver or fatty fish. And the starting molecule is retinyl palmitate. The palmitate creates an ester, and this is the way by which you can stabilize the molecule. If the ester is gone, so you just go into the alcohol, then you get a retinol, and retinol is much more reactive. So it means that, for example, if you would organize a supplementation campaign, you would always use retinyl palmitate because that one is stable in the capsule. If you would put it as retinol in the capsule and then start distributing it, for example, in a tropical area, you're pretty sure that most of the activity will be gone before the target person takes the tablet. And so you need to stabilize it, and this is done in the ester forms. For the plant-based foods, we mainly have carotenoids. Carotenoids are provitamins, so it means that they have the capacity to become vitamin A, but in order to be like that, they have to be converted in the human body. So during the metabolism, these carotenoids can be converted into vitamin A, which can then be used in human metabolism. These carotenoids can be found in green, yellow, red vegetables. The most classical one known is, for example, carrots, who contain beta-carotene, which creates the orange color of carrots. But they are present in many more vegetables. And think, for example, about spinach, which is a very green vegetable. But the green is caused by the chlorophyll. So if you remove the chlorophyll by extraction, the color will become orange, yellow, red, and these colors are actually linked to the carotenoids. So the carotenoids are physically there, but when chlorophyll is there, it will mask the normal color of the carotenoids. Also keep in mind that not all the carotenoids are vitamin A active. So in order to obtain vitamin A activity, you need to have a specific ring structure, which I will also show you if I show you beta-carotene. If the ring structure is not there, then the molecule does not have vitamin A activity. A good example is lycopene. Lycopene is a deep red color that you can find in tomatoes. This lycopene is a carotenoid. It has the same similar intermediate structure, but it doesn't have the ring structures. So it can't produce vitamin A in the body, although it is a carotenoid. The beta-carotene, the orange color from carrots, does contain vitamin A activity. So in principle, whether it is an animal or a plant-based foods, once they enter in the human body, during the GI tract digestion, retinol is released. This is done by hydrolases, and then the free molecule can physically be absorbed. After absorption of beta-carotene, the carotene is converted into retinol, but the yield is not 100%. And this creates a lot of confusion also among students, so be a bit careful with this. In principle, the conversion has a yield factor of 50%. So it means that if I convert one molecule of beta-carotene, which theoretically contains 2 molecules of retinol, I'm going to lose one molecule of retinol. So only one of them is left. The final released retinol is being absorbed, and the absorption yield is around 33%. So it means if I have one retinol equivalent as unit, so 1 micrograms of retinol, then I know that this is equivalent to 6 micrograms of beta-carotene. And because 6 micrograms with a yield of 50% is 3 micrograms, and with a yield of 33% is 1 micrograms. So the conversion is 1 out of 6 to go from retinol to beta-carotene. And if you would take the other carotenoids that are still having vitamin A activity, we even think that they are lower in conversion yield. So there the proportion is 1 out of 12. And if you look into the articles, then we can even say that there's still some discussion about what is now the ideal conversion factors. I'm saying here 1 out of 6, 1 out of 12. Some scientists actually agree or indicate that it should be 112 and 124. So much higher than what we actually indicate here. So the retinol equivalent is a unit that you can use in case we are talking about foods, because there's an absorption yield and you have to release it from the matrix, so the yield is lower. If you give it as a tablet, so in pure form, then you get the so-called international unit. And one international unit is 0.3 micrograms of retinol, equivalent to 0.6 micrograms of beta-carotene and 1.2 micrograms of other carotenoids. So in this case, you can see that there's still a yield factor of 50%, which is related to the conversions, but there's no yield factor for absorption anymore, because it's a supplement and we assume that it is already free from the matrix, so that it is much better bioavailable than what you would get with a classical food product. So here you can see this conversion, so from beta-carotene, so you can see the structure of the molecule on the top. So it's a long chain molecule with all types of double bonds, and on the left and the right side there's a ring structure. If the ring structure is gone, like in lycopene, then the vitamin A activity is gone. If the ring structure is there, then you can keep your activities. So an oxygenase will normally cut the molecule into two. You will get retinal, so who is still an oxidized form, and the retinal will be reduced into the retinol. And the retinol is the free molecule that can be transported, but it's not necessarily the active molecule. And so you need to activate it. I will show you that immediately in order to make it active in the human body. So once the absorption has taken place, then it can be transported. This is done by the so-called retinol binding proteins, RBBs. And the C stands for cytoplasmic or cellular. So it's usually transported first within the gastrointestinal wall up to the bloodstream. Then it goes into the chylomicrons. It's a fat-soluble carrier, which we already saw in the chapter on lipids. So the chylomicron will also absorb the retinol, and the retinol will be transported as an ester, so the palmitate, for example. It will go to the liver, where it will be stored in the stellate cells, and from there it can be released and transported again, in this case with a plasma retinol binding protein. Finally, it goes to the target organs, and there it can be taken up, for example, in the eyes, where it can again bind to specific retinol binding proteins, again, in this case, cellular retinol binding proteins. So you have transporters within the cell, and you have transporters within the blood or within the plasma. It's very important to see that if you want to transport vitamin A, you need protein. If a person is very malnourished, with a very low protein status, the person will automatically also become vitamin A deficient. not necessarily because vitamin A was not there, but especially because the transporters who have to bring the molecule to the right target are physically not there. So you can't transport vitamin A and you become vitamin A deficient. Once it reaches the target, it has to be active. And there are several types of activities that we know today. So the first one is the conversion of retinol into the holo form. And holo means that you bind the vitamin A to a protein. In this case, the transporter, which is the retinol binding protein. If they are both linked together, then you have the holo form. If it's not linked together, so you just remove the vitamin A, then you get the apo form, and then you get the apo-retinol binding protein. So think about this, because if you want to determine some ideas about vitamin A status, you might think about, trying to do this with this type of holo apo forms. So retinol can be converted into the ester, we already discussed, so this is useful for stability. And then it can also be converted into a glucuronide. And the glucuronides are important for release of vitamin A in the GI tract, because this is done by bile acids, so by bile who is going from the liver to the gallbladder and from the gallbladder to the small intestine. And why is this important here? Because if you have an overload of vitamin A, for example, you would take tablets at 3-4 times the maximum allowable dose, this vitamin A cannot be released from the human body. You can't remove it by the urine. It stays stuck in the body, usually in the adipose tissue. And in order to get rid of it, the only option you have is going by the gallbladder. So in this case, it goes to the GI tract, and from the GI tract down to the colon and into the fecal material. It's not the ideal route, but it's one of the few routes we have to remove excess of vitamin A. Then the fourth route that you see there is construction of glycoproteins, whereby combinations are made between sugars and proteins. And in order to make that connection possible, we need retinol. So retinol will activate the sugar, and then the sugar will be incorporated into the protein. And then we have the last reaction on the bottom of the slide, and this one is the most important one. So you can mention that in your notes. This is a very important reaction because the retinol will be oxidized to retinal reversibly with NAD, and the retinal will be oxidized to retinoic acid, which is an irreversible process. And it's the retinoic acid, so the molecule on the bottom of the slide on the right, this one is the active molecule. So this one is the hormonal type molecule that will go to the nucleus, that will interact with the receptors of the genetic material, and there you get translation transcription reactions based on the coupling of retinoic acid with genetic materials. If retinoic acid is not there because of vitamin A deficiency, then you get disturbances which can lead to eye deficiencies, which we call xerophthalmia. So I hope that is clear. The activated molecule, which is a typical example of a bioconversion, is retinoic acid. You don't have to know the chemical formula, but I do want you to know the word. So you need to know the different forms: retinol, retinal, retinoic acid, retinyl esters, beta-carotene. So the words need to be known, but the formulas behind it do not have to be reproduced. So I come to this very important function of vitamin A. And so in this completely related to the systemic hormonal function of retinoic acid. So when retinoic acid can bind to the cell nucleus or can interact with the cell nucleus, then you get tissue differentiation and growth. So the growth, the renewal of the cells, at the level of the eye cornea is actually determined by retinoic acid. If this doesn't occur, then you get in the first phase what we call reversible night blindness. So it means that when you are in a gray area, let's say it's not yet dark, but it's already quite gray, and in those conditions you have difficulties to see and to distinguish objects, then you are in the phase of reversible night blindness. It's still reversible, so if you would get vitamin A, you would improve your sight in those gray areas. However, if it's not corrected, then you go into the irreversible keratinization of the cornea. And the cornea is on top of the eye bolus, and this cornea normally on a regular basis is renewed. And so the cells are multiplying, and you always get new cells that produce new cornea. But if the vitamin A in the form of retinoic acid is absent, then there is no renewal of the cornea. Then there is keratinization of the cornea surface, and then you get what we call xerothalmia, which is actually irreversible blindness. And this occurs with young children below the age of 5 years, especially in developing countries. So as a result of this very strong relationship, today, young children below the age of 5 all receive vitamin A tablets in order to avoid that the xerothalmia can break through. But of course, you can understand that giving tablets at such a large scale is definitely not a sustainable solution. And so you have to think about new ideas on how to replace vitamin A supplements by ordinary dietary patterns. So what is now the mechanism? So retinoic acid goes to the cell nucleus. It binds to receptor proteins, which is, for example, retinoic acid receptors. And there it leads to the synthesis of polyamines. There is an enzyme involved, which is called ornithine decarboxylase. But once the polyamines are synthesized, the cells start growing, start multiplying, creating new types of cells, and in this way you create new cornea. As a result of that, you get an increased transcription of the ornithine decarboxylase, which is simultaneously done in the presence of estrogens. So if estrogens would not be there, then the mechanism is quite clear. You have retinoic acid leading to polyamines, and the polyamines are important for the growth of the cells. If estrogens are there in addition, the story could be a little bit more complicated because they could also simultaneously interact with the ornithine decarboxylase. But I hope with this you have already an idea what is the link between growth of the cells and the presence of retinoic acid. So the question is this now important? I already mentioned it to you. Yes, it is. We know the vitamin. We know there is a deficiency. We also know the implications of that, which is mainly this irreversible blindness. So if we look on the side of the World Health Organization, then you can see here in the colored countries, the countries that were at risk, especially around 2005. So I would say about 20 years ago, we had this specific situation, whereby you can see that the dark green or even the dark gray zones are the zones where vitamin A deficiency is quite prominent. And so we're mainly talking about the middle part of sub-Saharan Africa, and we're talking about some specific isolated countries, like for example Bolivia, who is located in Latin America, and also some countries here in the Southeast Asia. Some countries are really dark green, and dark green means that they are severely vitamin A deficient. For example, here, the country in the northern part of Africa is Sudan. And you know that there is already a civil war going on in Sudan for many, many years, which automatically also means that it's very difficult to enter into the country and to sustain the campaigns that distribute the tablets. Yeah, so that's a big problem with the current networking. You can reach the target groups, so you can reach the children. But the problem is if the area is unstable, and there's especially war region zones, it's getting very difficult to get the tablets to the persons who really need it. And so it's absolutely not a sustainable approach. Now you will probably ask me, what is the situation today? So in order to help you with that, there is here a website that you need to check. Is the website of UNICEF that actually gives you the most relevant information. So I'm going to try to open it, but I'm not sure if it will work within the program. I assume that you cannot see this. So what I'm going to try to do is to share my screen in the hope that it will work. So I hope that everybody can see this. So this is normally the website of UNICEF that will give you some information about the recent data for vitamin A. And as you can see here, the updates are around 2023. So it is actually for this year. And I will just show you with a land graph some information. So here you can see that in 2022, nearly 2 in 3 children in need were protected by giving 2 high-dose vitamin A supplements. So you don't have to give it every week or every month, but the supplements that you need to give contain about 200,000 international units in one supplement of vitamin A retinol palmitate. And this one is supplied twice a year. So one tablet can actually protect the child for a total period of 4 to 6 months. So they get 2 a year, and that is normally sufficient to avoid development of xerothalmia. So in 2022, 2 out of 3 children in need were actually reached, so that's already a very big success. So if you go a little bit to the mapping, then you get a better idea on how it really looks like for today. So on the map, you can see the countries that actually received the supplements, and there's a color code which also indicates that some countries are quite easy reachable. So it means that, for example, more than 80% of the children did receive the supplements. But if you go more into the purple colors, then you can see that even less than 20%, so less than 1 out of 5, reaches or is reached by these supplement campaigns. And in Africa, for example, you can very clearly see that some countries are quite successful and some other countries are totally not successful. The reason why there is such a big difference between the countries is, of course, a more political question. Because why is it that Sudan, for example, is not successful and then the countries neighboring it are quite successful? Well, the major reason is, of course, stability within the country. If you get a stable regime, then in principle you can aid in the distribution. If the regime is less stable or more difficult to access, in those situations it will be more difficult to get the tablets to the place where they are needed. So if you want an update, you can just go into that website, and there's a final thing that I wanted to show you from the website that is interesting to know. So if you go a little bit further down, you can open here, read more. And then you get these graphs. If you can, you just look at these graphs because it's quite interesting. So here you can see for several areas in Africa. So here is those West and Central Africa, East and Southern Africa, South Asia is also here, and then some other least developing countries. So what you can see here is the coverage with vitamin A supplements given twice a day as a function of the years. And it starts in 2017 and it ends in 2020 for 4 different regions. And what here is just the overall countries, everything together. So what you can really see if you go from left to right is that in all these regions, you see a drastic drop around 2020. And it starts more or less in 2019 and it continues very drastically in 2020. And you probably know what is going on here. Because in that area, in that period of time, we had a COVID crisis. And during the COVID crisis, we saw that all the connection lines that were normally installed in order to have help facilities to, for example, children in rural areas, and that is not only vitamin A, because could also be vaccine campaigns or whatever. You saw that during the COVID crisis, this was drastically reduced because all the connection lines were gone and there was no transport possible. There was no interaction anymore with the different regions, the target regions for vitamin A. So also the vitamin A supplement campaigns dropped drastically. And this is a very clear indication that although we have a solution for vitamin A, it is definitely not a sustainable approach. And because as soon as something like this political, or in this case, political or medical crisis occur, immediately the system falls down. So we really have to think in the future about new methodologies to replace the current campaigns into a more sustainable and more resilient type of campaign. So I hope with this information you have some latest data on how it looks like. So I'm going back now to my slide presentation. Here it is. So we know now more or less what the mechanism is by which vitamin A can interact with xerothalmia. We know the current situation, and we know that we have some problems still with sustainability and resilience. So if that sustains and if there is no successful campaign, the first thing that happens is the so-called Bitot spots. And so you can see that here in the eye bolus, which means that the cornea, which is the outside layers of the eye bolus, is actually drying out because there is no multiplication, no renewal of the cells. So you get these first already clinical signs that can lead to eye blindness and that need very urgent intervention. If that intervention is not there, then this leads to so-called xerothalmia, which means that the eye is actually completely lost for sight. So it's an irreversible eye blindness. The cornea is damaged. And since this is very young children, it means that this is blindness for life. And so that's why it is so important to sustain and to maintain actually these vitamin A campaigns, even if they are not sustainable on the long term. Next to this first function, there's also some other types of functions for vitamin A. The first one is actually coming from the animal nutrition research, whereby it was seen that if you dry forages, you will of course lose your beta-carotene and your vitamin activity. And this leads to problems with immunocompetence and reproduction. And this was mainly the case with production animals. And this was mainly linked to retinol. And so not anymore to retinoic acid, but this effect was linked to retinol. So in the human nutrition area, when the results were known about effects on reproduction, they started to think that if you give supplements to pregnant women, that it may help in the birth outcome, because there's a link between vitamin A and, in this case, reproduction. So they did that. I think it was done somewhere in the 1970s. And it was found that if you give high levels of vitamin A in the form of supplements, and you give it in a chronic way during pregnancy, that it can become dangerous. So that's why nowadays, based on this study partly, we now have maximum tolerable intake levels for vitamin A in the form of tablets. You have to respect it, certainly if you are pregnant or willing to become pregnant. A second example, which was more recently in the 90s and the beginning of 2000, there were large intervention trials which were done with supplements of beta-carotene. And these were organized in Finland, I think, in UK, in the USA. So simultaneously, several intervention studies were running, whereby the supplements were given to smokers. The idea was that beta-carotene is an antioxidant. Smoking creates radicals. If you give them antioxidants, you might reduce radical formation, and this could help in avoiding that people would get lung cancers due to smoking. So they gave the study, they did an intervention, which was a randomized controlled trial, double-blind, so control group and intervention group. Nobody knew in which group you were actually located, but when the study was almost over and they actually opened the key, they saw that people who received the supplements had a higher incidence of lung cancers. So it means that this is a negative effect, especially during smoking. We now know the mechanism. We know that the smoke particles who are residing within the lung tissue can be activated by the physical presence of beta-carotene. So beta-carotene in those situations is not an antioxidant, but it becomes a pro-oxidant. So it can induce radicals rather than reducing them. So these campaigns were immediately stopped, they were blocked, there was no possibility anymore to give them. It again illustrates that taking isolated samples of antioxidants like beta-carotene or vitamin A, you put them in a tablet, you give them in high doses, they could become toxic. So you really have to respect the maximum intake levels that are now defined. And then the final potential mechanism or function that it has in the human body is light sensitivity, whereby retinol is oxidized to retinol and retinol is reduced again to retinol. And this creates the eyesight that we have today. So the black and white that you can see, the colored formation that you can see with the eyes, all these elements is determined by conversion of retinol to retinol. But this part is not the most important one. As I already said, the most important mechanism is the interaction of retinoic acid with genetic material that induces proliferation, so multiplication of the cells of the cornea of the eye. Okay, and this leaves us for about 10 minutes before we have the break. And this makes us ready for the final problem-based learning question. So I already told you last time that we will do one problem-based learning question in class, and we will do it in a slightly different form than we did it in the past. So I will now give you a question. I will ask you to solve it alone, so no discussion with other people. You have to do it yourself. And your time frame is restricted to maximum 10 minutes. Because on the real exam, you also have to work alone, not with your colleagues. You don't have your direct course notes, and you only have limited time. I guess you will have about 10 minutes time to solve a problem-based learning question on the exam. So we're going to try this out to see if it works. I will give you 10 minutes to solve the problem, and then we immediately discuss it in class. So the question is the following. So take something to write, please. Just a blank paper. Take a drawing pen. And if you can, write the answer in words on the paper, because that's how you're going to do it on the exam. So if you want to really feel a little bit if you can fit the time frame, just write it out in words, no formulas, just how you see it and how you answer this question. Okay? So if everybody's ready, that I'm going to give you now the question. So the question is on the determination of the vitamin A status in humans. And on the slide, you can see on the x-axis the weeks that experimental animals, which in this case are rats, are receiving a diet which is free from vitamin A. So they don't receive vitamin A by the diet. And they are consuming this diet for about 8 weeks. Every week, we take samples and we measure vitamin A, just to get an idea about the status. On the y-axis, you can see the percentage of the normal level. The normal level is, of course, 100% at time period 0. And so just before starting the experiment, the rats are normal. But then as a function of time, they are actually losing vitamin A because there is no vitamin A physically present in the diet. So you get 3 curvatures. One of the curves is linked to the blood. So samples taken from the serum, the plasma, or the blood. One curve is linked to a functional test, in this case to eye deficiency. So you're just going to see if the rats can still have normal eyesight. It's a functional test. And the third curvature that you see is a curvature from the liver. So you have 3 curves. One of them is the blood. The other one is a functional test in the eye. And the third one is the liver. So the question is quite clear. Tell me which curve is what exactly. And of course, explain this. And then, based on that result, tell me which analysis you're going to do in order to determine the vitamin A status of the rats. And so which analysis of the 3 is the most optimal? And of course, why? So you see 3 curves. The first curve on the left side, which is the black curve with the black dots, this is actually the liver, because the liver is, of course, the first organ that will lose vitamin A as soon as there's no vitamin A coming in from the diet. So the liver is actually compensating for the deficiency. And this also means that the 2 other curves, which are linked to the bloodstream as well as to the eye functionality, will not show any significant difference. As soon as the liver is completely replenished, which is happening around 4 to 5 weeks of vitamin A free diet, then of course you will see that the bloodstream, which is the second curve, the dotted line, you see that it is significantly reduced in a very short amount of time because the liver cannot compensate anymore. And then as soon as the blood will also lose its vitamin A supply, in those situations you will see that the third curve, which is the open white dots curve, there you see that the eyesight will also go down, which is a functional test, which is not so sensitive because it takes quite some time before you will see something with that test. So although it can be useful because it has not too much ethical restrictions, it has the least sensitivity. So in this respect, you can only use it if you are fine with the idea that you will only see effects when vitamin A deficiency is already drastical. So since liver is not an option because then you'd have an invasive technique, which we cannot do with young children, we're left with the blood, which is probably the most optimal way to work on it. But you have to take into account that in the blood, the drop could be quite significant in a very small time frame. So you really need to be at that borderline in order to have a significant detection of vitamin A status. So I hope this gives you more or less an idea on how such a question can look like. You can perfectly answer it in words without having using of formulas or complicated graphs, but the answer does have to be complete. So you need to give me a clear view, motivated also why you would go for a certain choice, and you have to explain as much as you can from what you see on the graph. Okay, so I hope with this you have enough information to prepare yourself for problem-based learning questions. So moving on into the field of vitamin A, then the question is, what are we doing now in the field? So I already explained to you that today we have large-scale supplementation campaigns. They are supported by the Helen Keller International Foundation. Helen Keller was actually a blind lady who was living in the States and who actually sacrificed or gave her life to do as much as she could for blind people in the world. And one of the initiatives which was taken in there was also campaigns to avoid eye blindness with young children in developing countries. So part of the current network that we have today is still supported by the Helen Keller Foundation. Of course, it's not sustainable. We already discussed that. So we come to the second level of intervention, and that could be the use of interfering with farmer production. So there are projects running in Africa, for example, in Uganda, where they are working with improved sweet potatoes. So these potatoes have an orange flesh internal part, and this is of course carotenoids that could have vitamin A activity. So the idea here is that you actually stimulate local farmers to produce more sweet potatoes, then to convert these sweet potatoes into different types of food products, which could be acceptable for the local communities. And then you also finally stimulate the local communities to start eating more sweet potato. If we could get that done on a larger scale, then it could be an alternative for the use of supplements. And it could also be more interesting because perhaps, I'm not 100% sure, but it could perhaps have a higher resilience and a higher sustainability. And because you don't have to go into the communities to supply the supplements, you can just produce it in the communities as such. So as long as there are no severe warfares going on, or there is a normal way of living with people having the opportunity to harvest their crops on the field, in those conditions, principally speaking, the sweet potato could form an alternative. And then the third option, which is also quite heavily investigated, is the use of biofortification. So in this case, we talk about golden rice. You might know the product. It has been produced by the Bill and Melinda Gates Foundation. Support was given for that. So the golden rice is actually a yellow type rice, which is enriched with beta carotene. So it has been genetically manipulated in such a way that the levels of beta carotene are systematically increased. And when people eat rice as staple, in this case, they could also have higher intakes of vitamin A. This is already positively approved in Canada, United States, in Australia, New Zealand, several areas in the south. But of course, as you know, it is not approved in Europe because Europe does not want to work with GMOs, genetically manipulated foods in this case. So it's mainly restricted to the areas where this genetic manipulation technology has been officially approved. So I hope with this you have a good idea on the different options, their advantages, their disadvantages, and their potential options for the future. A final slide on vitamin A is actually linked to the Global Alliance for Vitamin A. So today, all the initiatives that are actually taken around vitamin A are more or less grouped together by the Global Alliance for Vitamin A. They have a website, so you can just go there. It's called GAVA, and it will give you all information about strategies and things like that. On their website, they also refer to a recent report which was made by the World Health Organization. You can see the title page on the top about vitamin A supplementation in infants from 1 to 5 months of age. And on the left side, you can see more or less the basic conclusions. So today, with the supplementation campaigns, we have tablets of 200,000 international units given to children of 12 to 59 months of age, and it should give them protection for 4 to 6 months of life. And so it means 2 of the supplements a year is enough to protect them. These supplements are well tolerated. It seems that they can give some side effects, but it's only in a minority of the children. One of the examples is diarrhea that sometimes occurs with some children if they receive the tablets. But generally speaking, for a large-scale intervention, the number of side reactions is quite limited. They're supplied in the form of gelatin capsules, and they cost about $0.0 for each supplement, which means that if you can avoid xerophthalmia, you can also avoid that there should be a medical intervention, which means that you have a cost gain of about $200 to $250 for each supplement which is supplied. And so it's very clear that although it is an expensive campaign and difficult to organize, they still keep it running because it definitely has effect. And in terms of cost price, it's also, let's say, financially beneficial.